Hi guys, and welcome to Mindset Learn's Learn Extra live shows. As you guys already know, we are tackling life science today, and Kathy is with us. And I know, well, Kathy's with us through grade 10, 11 and 12.
3 hours.
3 hours, both of us, we're here. So guys, please, we're doing this for you, so stay tuned, we're going to do some amazing stuff. Kathy, today we're going to be talking about cells, right?
That's right. Cells for grade 10, and people, it's the basics of everything you're going to do in grade 10, 11 and 12. So pay attention, learn now, and everything else will just fall into place.
So hopefully there's some grade 12s watching too, right?
Definitely.
Fantastic. We're going to take a short break and we'll be back with you now. Bye. Hi guys, and welcome back, and are we excited for an amazing lesson. Join us on Facebook, facebook.com/learnextra, that's XTRA, also on Twitter @learnextra. I am so excited about this lesson. I know Sisanda even Facebooked us right now, and she said that she's in a taxi on her way home from school and she can't wait to get home so she can tune into Mindset. Sisanda, I hope you've made it. If you have, let us know. So I think let's get straight to it. Kathy, we're chatting about cells.
Okay, people. Hi, everybody. You would have noticed this, and this is a microscope. Now, if something is micro, it means it's very little. If it is macro, it means it's big. So micro would be a microscope, which means to see or to look at. And you would look at microbes or tiny little things that you cannot see with a naked eye, which is pretty much 90% of what is on our planet, believe it or not. All right, so we are going to focus on cells. And, okay, let's just do our microscope here. What I've done is John has put a slide of the microscope on here for me, and I just want to get a different pen color, so we're going to use red. And okay, we've got the microscope. If we look at the actual microscope, you've got a tube-like structure, and that tube-like structure is called the body tube. Now that's not really hard. Then at the end here of the body tube, in other words, this structure along here, we have a little lens. And that little lens is called the ocular, because your eyes, everything to do with your eyes is oculomotor muscles, and the whole vision process is here because of the eye, ocular. All right? So here we've got an ocular lens. Or, if you want to really make it easy, you can just call it an eyepiece. All right. But that ocular lens has a magnification. I hope you can see the ION on the black. But it has a magnification as well. It can be anything from 5 times to 10 times. So it is able to magnify whatever you are seeing. All right, now, looking at this little microscope, and I'm going to take you to our little thing over here, what we've got is this part here, and that's where you carry a microscope. Even if it's one of those huge proper microscopes that you'd have at school, they're heavy, they're big. You would then hold it on the arm, and you would put your hand below it, because if you drop it, whoa, it's expensive to replace. Okay, your parents are not going to be very happy with you. So the arm, we've got this tube, all right, which is the body tube, and we have the ocular, which is at the end. Now going down, we have this, there's your arm, which is the arm structure here. And then we have a revolving nose piece. Okay? Now the reason it's called a revolving nose piece is because when you are looking into the microscope, it's pretty much where your nose is going to be. So it's easy to remember to move this little wheel around like that. So it is the revolving, meaning turning, nose piece. And on that revolving nose piece, you have things that stick out. And those things are your objectives. All right. And the objectives each have a magnification power as well. So what we can do is we can say, right, you'll have one that will be 4 times, one that will be 10 times, and one that is 40 times. That is the common, or those are the common magnifications you will get on a normal school microscope. Okay, it's 4, 10, and 40. So if I wanted to look at something, the first thing I would do is I would put my microscope down. I would have the arm facing away from me, and I would set it to the lowest magnification. So you always start with 4, and then you'll move to 10, and then you'll move to 40. Okay, so you always start on the lowest magnification. Then you've got the stage. Now the stage is this little black structure over here, and on it you'll see clips, and what those clips do is they will hold your slide that will fit in over there. It holds the slide in place so it doesn't move around. We then have a coarse adjustment and a fine adjustment knob. Now, if we have a look at our little microscope that I've got here, it's clearly one, you know, from a little chemistry kit you'll buy in a shop. But the ones you'll have at school will have 2 types of buttons. The one that is higher and the one that your hand will move to while you are now focusing on whatever you're looking at on the slide, that is going to be the one that your hands will move to. So it'll be the one nearest and the closest to your hand. That is going to be coarse adjustment. So there you're going to focus. So its main function is to focus. And then when you want to focus extra carefully, you will then use the fine adjustment knob, which is sort of further forward. Now, the diaphragm's job is to allow light in. Because people, if you're looking at, anything that you've put, let's say I'm looking at onion cells, or I took some cells out of the inside of my cheek, and they're on a slide, if I want to see them, first thing I'm going to have to do is check them on the lowest objective magnification, and then I can move it higher. But every time I'm going to do coarse focusing, fine focusing, but you have to have light passing from below the slide through the slide, otherwise you're not going to see it. And to give you an idea, is if I took a slide and I put it against a black wall, I wouldn't be able to see the slide. It would sort of disappear. In order to see anything, we need light. So you can either have a mirror, let me smooth this down a bit. You can either have a mirror, which you would then tilt to any source, light source, okay? Or you can actually have a little button on the side here that you would click, and it would then shine a little bulb, or electricity would flow to a little bulb, and the light would then come up through there. But the amount of light that you allow through is due to the diaphragm. Let me just put my little circle back there. So, amount of light. Because you don't want too much light to shine through, because then you won't be able to see anything. It'll just be this bright thing, and it'll probably damage your eyes as well. You don't want a lot of light, you want just the right amount of light. So you would need to play with your light source, especially if you're using a mirror, and move your microscope to where the light is coming from. And you would then play with your coarse adjustment and your fine adjustment, figure the light again, and so you would carry on. For those schools and those of you that are lucky enough to work with light microscopes that are electronic, Well, it's very easy. Then you would use your diaphragm to control the amount of light that comes in. Now, in an exam, you would probably get a diagram of the microscope, and you would have to label the parts, or the teachers could ask you to, what is the function of point 1? What is the function of point 2? How does point 3 work? When would you use point 4? You must know the parts of a microscope, and you must know what their function is. It's no good knowing the labels, but I mean, if you use your common sense, it's pretty easy here. Okay. Have you got any questions, Indy?
Nothing yet, guys. Get them coming. Keep them coming. And Susanda, I'm so glad that you made it to your couch. So she is tuning in.
Did she make it home
Yes,
safely?
yes.
I sometimes wonder with those taxis, hey? But one thing I must say is they know exactly to the millimeter how big the taxi is. You know, so they can
squeeze into small places.
Sure. I don't know. I think my nerves would be finished. I certainly wouldn't be able to take a taxi here, Indiana, on the way to our live session here. I'd be a nervous wreck. All right, people, here we go. The plant cell. Now, you must learn the plant cell and the animal cell. But what is more important is that you need to know each structure in a cell and what their function is. Now, before we move on to the actual diagram, I just want to show you a basic concept. There we go. Is we start off with cells. Okay? And the little cells, well, a whole bunch of them, group together to form tissue. So it would form one layer of tissue. And a whole lot of tissues group together to form an organ. I must be careful of not having my hand anywhere near here. Organ. And quite a number of organs will work together to work as a system. And all the systems in the body coordinate and work together to form an organism. And we went through this, remember, last week if you were watching. We have cells, lots of cells together form tissue, lots of tissues form organs, an organ. Various organs will form a system, and the systems together make you. So let's look at an example. Let's get another color here. Okay, cells, let's say, I'm trying to think, cardiac cells. Okay, cardiac should tell you that you're going to find them in the heart. Okay, so your cardiac, all your different little cardiac cells will join together to form the tissue that makes up the heart tissue. Okay, and that heart tissue will end up joining to form the heart. Right? And the heart plus the lungs plus the heart plus the lungs, I'm trying to think, plus your blood vessels, so your arteries and your veins, all join together to form your cardiopulmonary system, and that supplies and pumps oxygenated blood and to the heart and to the rest of the body, and carbon dioxide away from all the organs, and it makes sure that our organism is fine. So we may have the circulatory system. Okay, and you'd have the respiratory respire, to breathe, respiratory system. You have the digestive system, and they all are made up of cells that group together to form tissue, that groups together to form an organ, that groups together to form a system, that groups together to make us an organism. In other words, everything is coordinated and works properly together. And that is the function of the brain and the nervous system. All right, so, which you're clearly going to do at a later stage. So now, having said that, we've got two types of cells in the body. Two types of cells that you are going to do. You are going to go through a plant cell and its basic structure, and the animal cell and its basic structure. These are not specialized, these are general structures of the cells. Okay, but for example, when you get further on in the FET phase, you are going to learn, like for example in grade 11, you are going to do the different types of cells for animal and plant tissue. So you've got the xylem and you've got the phloem, which transports water and nutrients up the plant, and the phloem, water and mineral salts, and then the phloem that transports water and dissolved nutrients up and down the plant. You're going to have those little root hairs. You're going to have stomata in the leaves. All these different cells that help those organs to function. In us, you've got, for example, epithelial cells, which line any structure that is exposed to anything that either goes out of the body or comes in the body. So you'd have squamous epithelium here in your mouth and in your rectum, for example. You would have cuboidal epithelium in your urinary tubes. You would so it carries on. I'm now telling you different types of cells, and maybe when it gets to that section in grade 11, you'll think, oh, hang on, we did that, I know that. But just remember squamous cells in the mouth. They are the most basic epithelial cells you can get. Then you've got striated muscle cells, which is your skeletal muscles, which you do when you do the skeleton in grade 11. When you go to grade 12 and you have to do the reproductive system and you have to do the nucleus and the whole process of genetics and nucleic acids, you have to know the basic functions of cells, people. So we're going to talk about a cell. Now remember, inside any cell, I need actually a blank page. So I'm going to do this and see if I have any blank pages here. Yes. Okay. Oh, let's try this one. There we go. You have a cell. Inside that cell, we have little structures called organelles. So, people, we're going to go one step further. We're going to say, right, we start off with organelles. The organelles make up a cell. Lots of different cells make up tissue. Lots of layers of tissue make an organ. And many organs make one system. Lots of systems make the organism. And that applies to plants and to animals. So now we are going to be looking at the cell. And there are two types. Okay? There's plant and animal. So plant cells, you will find, are always classified, or any classification as a plant cell means it has a cell wall. That's what makes a plant cell what it is. And different to an animal cell, because an animal cell never, ever, ever has a cell wall. Now, the cell wall in a plant is made of cellulose, and it is able to assist the plant in helping it with its structure. Okay? So, here we have our plant cell. And I'm going to show you a line diagram, and if I ask you to do anything, learn this, okay? You will, in any test and in any exam, get either an animal cell or a plant cell that you would need to label parts of. You would be asked about the structure versus the function. So, think about this. Structure and function. If I have something and it has, okay, if I had a glass and a plate, okay, would I put my dinner, let's say I have meat and potatoes and vegetables, okay, potatoes and vegetables, so meat and vegetables, would I have my meat and vegetables on a plate, or would I stick it in my glass? And you're all laughing and saying, oh, for heaven's sake. Well, those are the kind of errors you make in class because you don't think. It's structure. The flat plate is structured to suit food. And the glass or container, long cylindrical container, is designed and structured to suit my liquid, because I can't put liquid in a plate. I'd have to lick it up like a cat or a dog. Okay, so everything you do from now to grade 12 is structure versus function. If a thing looks a certain way, whatever the structure is, it is specific because it is to perform a function. All right, so any fold that you see anywhere in any structure will tell you if there is a fold, it increases the surface area. In other words, the area that is exposed to the surrounding sides. All right, so simple things like that. Those are basics that you need to know. You must know your definitions. And this goes for the grade 10s, the 11s, the 12s, the grade 8s, and the grade 9s, same thing. In life sciences, learn your diagrams so that you know the structures, and learn the function of those structures. And if you know that, you know your definitions. So learn your definitions. All right, so here we go. The plant cell. Now you'll notice this light green structure all the way around. And that light green structure is what we call, okay, I'm going to need a color here, otherwise you're not, and I'm sitting with this red. Here is the cell wall. Now, cell wall is rigid. It's made of, heaven's sake, cellulose, and the cellulose is a carbohydrate. It's a polysaccharide, and it gives the whole cell, and therefore the plant, it provides it with structure and support and shape, because this is rigid. So this cell can't like billow out here or have a rounded shape unless the cell wall is rounded. So the cell wall provides structure versus function. The cell wall is made of cellulose, you must know that, and it provides structure, shape, and support to the plant cell. Very important. And in animals, there are no cell walls. Okay, only in plant cells, not animal cells. So that's one difference already. Then we have this structure here, which is right next to it, and I'm going to do this, and that is the cell membrane. Now, plant and animal cells have a cell membrane, and pretty much, if you are going to talk about anything moving into a cell or out of a cell from now until you are an old person with regards to this subject or any biological or science field, you must know that this cell membrane is a shared characteristic between plants and animals. And we can link that back to evolution, where they say, and the theory is that it's one of the theories that they say backs up the fact that we all came from some little explosion that happened in the middle of the sea, all right? And the organisms became more and more and more complex. But the fact that animal cells and plant cells share cytoplasm, they share nuclei, they share vacuoles, they share cell membranes, all points to a commonality between plants and animals. Now remember, it's a theory. That's all, the whole diversity, evolution, blah, 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 all of that is purely theory. No one can prove it. Nobody lived 500 million years ago. All right, so it's just a theory. But it's one of the things that they say, or one of the aspects that they say, tends to point to the fact that animals and plants had the same origin. All right, so the plant cells, they develop cell walls, and then you've got your cell membrane, which sits right against the cell wall. Okay, right up against the cell wall. Now, I need, hold on, let's do this.
And you were doing so well, Kathy.
Hey, listen.
Hey?
We're getting there.
No, she's got it. She's got it. I'm only teasing her.
I'm just going to, let's go to this one, and I want to change it to purple. All right. Cool. Done. I still haven't done it.
No, it's okay. Do you want me to? You know
Very
what?
difficult to find the black background. Okay, let's go with this.
Okay, while you're doing that, Kathy, I just want to ask mindsetters, please, 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 guys, let us know if you have any questions, but try and make the questions relevant to what Kathy is talking about. It'll be so much easier. While we're chatting about cells and all these things, say, hey, I don't understand this, I don't understand that. Let's try and stay on topic. Makes things much easier. Guys, and I hope you're having a really awesome session. And I think Kathy has worked out the board, so we can go straight back to her. Hey, you ready for us?
Definitely.
There you go.
Got
hey?
it. Nice, bright.
No,
it looks as you can
you've
almost
got it.
imagine you're in class.
No, you've got it.
We've got the cell membrane. People, you must know this structure because it'll make sense to so much more that you are going to learn. All right, the cell membrane, first thing, is that it is partially permeable. Okay. In contrast to the cell wall, because the cell wall is completely permeable. All right, that cellular cell wall is there purely for structure and support. Its job is not to check and guard what comes in and out of the cell. That is the function of the cell membrane. Its job is to say, no, 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 no. You are too big, you're not coming in. Or you are wrong, you're not coming in. And that is what it does. Think of the cell membrane as the policeman of plants and animals. It's what makes sure that all our organelles for the cell are in place, and it makes sure that, listen, only the things that belong there are in there. All right, so it's partially permeable. Some teachers will say semi-permeable. Okay, some will say differentially permeable. They all mean the same thing. All right, now, permeable means allows certain substances through into the little cell. Okay, and those certain substances must be a specific size. They must be small and dissolved, otherwise they are going nowhere. All right, everything moves with water in a plant and an animal body. So, here we go. Cell membranes partially permeable. Now, they have a structure that looks like this. They've got and then you'll have the same carrying on all the way around there, and you've got little structures like that. All right, and the same here. But here, hold on, let's take this one out. Let's make this a bit longer. Okay, now, what I've drawn here is your cell membrane, and it's called a mosaic. Okay, it's called a fluid mosaic, and this is what it is. these little guys, these little round ones here, okay, is, or this, hang on, let's just do this, let's make it simple. I'm battling with this pen. This is a phospholipid. And this is therefore made up of a fat, okay, a lipid, a fat, and phosphorus. And that is what makes the phospholipid. So you've got your phosphate sitting there and your lipids in between. And it makes like a double row of these structures. Okay, that's number one. Number two, this here and along here, that's protein. So that's a protein and this is a protein. And the two proteins form a channel into the cell, like a little passage into the cell. And that little passage is important because that is what makes the cell membrane partially permeable or semi-permeable or differentially permeable, depending on what term your teacher uses. I like partially permeable because it means it's sort of and not. It's partial. So whereas your cell wall would look like a roof that's held up by a whole bunch of pillars. And you've got these pillars that are sturdy and solid, made of cellulose, and 10 people can walk in and out of the structure. Whereas here, it says, no, 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 no. You are going one at a time through this protein channel. You are going, or substances, if they're very small, will be able to filter here through the double phospholipid layer. When we're going to take a small ad break, a very short one, mind you, and then I'm going to go through the difference between osmosis and diffusion. And people, please, you have to pay attention, get a piece of paper and a pen and write it down, because if you understand what I'm going to explain to you, you will be able to learn it, you will know it, and you will never ever forget it. It's that simple, but you must focus. All right, so I'll see you in, I think, 1.5 minutes. You have 90 seconds.
Welcome back, Mindsetters. We are so excited. We're having such a fab lesson, and I can't believe that we've actually only got 20 minutes left for grade 10s. It's ridiculous. You know what they say? Time flies when you're having fun. I'm telling you. Guys, Chabalala, we're going to get to your question. You know what it is. And I think the best way forward is just to let Kathy do her thing. She's on a roll, verbal diarrhea, going. Hey?
Thanks, Indy. You say the nicest things. At least you have my name right. Okay, people, here we go. The cell membrane, this is just a sort of a diagram to show you what I tried to draw. But remember, in an exam, you're going to have to draw it and label it, not something pretty 3D-like or 2D like this. But that yellow shows you your protein channel. Those are your little phosphates, and they're your lipids, and they form like a double layer. All right. Now, having said all of this, I'm going, oh, by the way, this website is amazing. You can see the most incredible animations and processes here on this website. So just have a look at it. It's KYRK.com, and on there you'll be able to pick up cell membranes, cell wall, nucleus, etc. It's nice to play. Alrighty, now let's make this go up. Osmosis and diffusion. Okay. Well, let's go with pink. All right. So we have osmosis and diffusion. You need to understand the difference. Okay. Diffusion is what I'm going to do first. Diffusion is something very, very simple. Okay. In that, with diffusion, it is just an equilibrium that is reached. Okay. So diffusion will continue until it gets to that equilibrium. So, for example, if I make a cup of tea and I put 2 spoons of sugar in, but I don't stir the tea, and I leave it for an hour, 2 hours, 3 hours, when I drink the tea, it's going to taste sweet. Why? Because the molecules of the sugar has moved around in the tea. Another one would be if I took air freshener and I sprayed just a squirt of air freshener in the front of a class. Who'd smell it first? Well, clearly me, but because I'd be the closest to it. But then the first row of learners, and then the next row of learners, and then the next, and then the next, and then the next. And as the molecules of the spray spreads evenly into the class. And if someone walked into the class, they would smell something really nice. So that would be an example of diffusion. It is when any substance moves from a high concentration to a lower concentration, okay, until an equilibrium is reached. So diffusion is the movement of a substance, whether it is solid, liquid, or gas, from a higher concentration downhill to a lower concentration. We say that it moves along the concentration gradient, okay, until an equilibrium is reached. I'm going to say that again. Diffusion is when the particles of a substance move from a higher concentration, so where there's more, to a lower concentration, where there is less, along the concentration gradient until an equilibrium is reached. Always an equilibrium. And something that is very important is that with diffusion, there is no membrane. Okay, there is no membrane at all. You can have it in the air, you can have it in a cup of tea, you can have it when you put chlorine in your swimming pool. All right, it is diffusion. High to a low concentration along the concentration gradient until an equilibrium is reached. In other words, until the high has spread so much to the low that it starts to tip up and even out like a seesaw. Now, osmosis. And osmosis is very, very easy. Osmosis would be if I had a hill. And over here I've got a chap. Okay, now please, I don't know, I'm not good at drawing. This little guy has got a skateboard. Now he wants to go uphill. This is low. This is low. This would be low. And this is high. Okay, so what happens? This little guy takes his skateboard and he walks uphill, which means he's going against the, I'm just going to do that, CONC gradient. Remember, we're talking about concentrations, but I'm using an analogy to show you. So he's going to walk uphill with his skateboard. He's going against the concentration gradient. He's going against the concentration gradient. He's going uphill, and he's going to need ATP. ATP is energy. All right. So anything that you read, for example, if you're a bit of a gym bunny and you look at all those supplements and it says it gives you ATP. ATP, or causes the release of ATP, ATP is an energy carrier. It's adenosine, which is a protein, with 3 phosphates. And the energy is in the bond between the adenosine molecule and the phosphates. So you'll have adenosine diphosphate, 2 phosphates. It goes to where energy is being made, for example, the mitochondria, because that's the function of the mitochondria. It is the powerhouse of a cell, and both plant and animal cells have a mitochondria. In fact, you have many mitochondria in cells. Okay, so you have the adenosine plus the 2 phosphates. It goes to where energy is being made, and the energy is used to combine that little phosphate onto the adenosine, the 3rd phosphate onto the adenosine. And it scoots off to where energy is needed. When it gets there, that phosphate breaks off, it breaks free, and that energy then goes to wherever it's needed. You must know that. All right, so we need ATP, we go from a high to a low, because you're going against the concentration gradient. But look at this one, and I'm going to use a different color. We're going to go from high to low. So once this little chap is up here, he gets on his skateboard and he goes whee all the way downhill. He's going along the concentration gradient. Does he need energy for this? No, he doesn't need any energy. Why? Because it's downhill. But he's going from what we call a higher to a lower what? Not concentration. He's going from a potential. His potential to be high to his potential to be low. So in osmosis, we don't talk about concentration, we talk about potential. And specifically, it is water potential. And the little sign for potential looks like that. So, in short, if we were to define it, we would say, I'm going to pick purple for this, that osmosis is the movement of, and we can't see this, so we go back to white. It's the movement of water, okay, only. We're only talking about water. From a high water potential to a low water potential, and it's going along that concentration gradient, okay? But you can never, ever, ever, and close your eyes and hear my voice, you can never have an equilibrium with osmosis. Not ever. All right, because the solutes that change that gradient cannot move through the membrane. They can't. So you can never have an equilibrium with osmosis. Whereas with diffusion, you can. So along the concentration gradient and through a partially permeable membrane. Okay, so listen. People, here we go. It is osmosis. Osmosis is the movement of water, not any substance, only water, from a higher water potential to a lower water potential, along the concentration gradient. Okay, will there ever be an equilibrium? No, through a partially permeable membrane. So, diffusion, movement of any substance from a higher concentration where there is more to a lower concentration where there is less, along that concentration gradient until an equilibrium is reached. Osmosis is the movement of water from a higher water potential, okay, because it's purer, it's got more potential, higher water potential to a lower water potential where things are mixed in. Along that concentration gradient, and it has to move through a partially or semi or differentially permeable membrane. If water moves anywhere and it doesn't go through the membrane, it is diffusion. What makes them different is that it is water moving through a membrane. That is what osmosis is. Now, quick, let's go back here because I still want to go through the plant cell.
Sure, we've got 2 minutes, eh?
Oh no, man, this is ridiculous. I need more time. All right, so with osmosis, that's what we have. But if we look at, I'm going to use lime green here, this part of my little hill, where we needed ATP and going against that concentration gradient, this poor little thing needs energy. And your molecule will have to be carried from where there is less to where there is more. And that would be for every cell in your body. everywhere. You could have any substance where you have more in the cell than outside the cell. Intracellularly is inside. Inter is between. All right. Inter, between. Intra, inside. So to get anything intracellularly, where there's more, we now have to make a plan. So it's like our little chap with the skateboard. He's going to go against the concentration gradient from a higher to a lower concentration. And you know what? This little skateboard is the substance. It must be carried. It's carried by our little guy. So for active transport, it's the movement of a substance from a low concentration to a higher concentration against that concentration gradient. And you need energy, and you need a carrier molecule. Then it is active transport. And people, active transport, osmosis, and diffusion is in every exam. Please learn it. And you know what? You get it again in grade 11, and you get it again in grade 12. As far as the cell is concerned, which I didn't even get close to today, I mean, I did the cell wall and the cell membrane, please learn the structures. The vacuole stores cell sap and water. Okay, the cytoplasm is where everything sits. Protoplasm is the nucleus and the organelles that float in the cytoplasm. So your protoplasm is the nucleus and the cytoplasm. All your hereditary characteristics are in your nucleus because that's where your chromosomes are. All right? And then you've got your vacuole, holds the water and cell sap. Plants have one big one, animals have lots of little ones. And remember, vesicles are any structure that holds another liquid within a liquid. So your vacuole would be a vesicle. All right.
Kathy, quickly, 10 seconds.
I've got
10,
no
9,
more time. I'm handing you over to Dearest here.
Yeah, you see, thank you, Dearest. She's in such a fluster because she's talking. Guys, I know that we've had so many questions today, and as you can see, Kathy is in a complete flap because she wants to teach you everything. She's such a fantastic teacher, and you know what? If you stick with her, I know for a fact that she will teach you everything you need to know for life sciences. Shabalala, I know that you had a question about the vacuole, but we are going to go over it next week. I've got my producer in my ear telling me so I'm going to save it, and we're going to go over it next week, and Kathy's going to be a very busy lady. Thank you so much for today, guys. Grade 11s, don't forget that you're on next. It's going to be a great show. Grade 10s, you've been fantastic, and we'll see you next week. Bye.